I'm Yulia Pavlova. I'm from Federal Research and Clinical Center of Physical-Chemical Medicine, and the topic of my presentation is the minimal in vitro model of G4 DNA in the chromatin context. G4 structures have been visualized in the genome of living cells and are prevalent in actively transcribed and nucleosome-depleted regions. They assist in chromatin structure rearrangements by recruiting, remodeling, and transcription factors. We aim to obtain a simple model system for studies of G4 impact on nucleosome positioning and density, and designed double-strand DNA constructs that enable direct comparison of nucleosome assembly on G4 versus non-G4 core DNA. First, we designed a DNA core sequence containing a nucleosome binding site and G4 or non-G4 linker DNA. The core sequence was assembled via kination and ligation of 55 base pair fragments. Then DNA was amplified and purified by extraction from agarose gel. We optimized histone octamer DNA ratio and assembled a mononucleosome using standard methods. Here, the yield of the mononucleosome was higher in case of non-G4 linker as compared to G4 linker. This agrees the assumption that G4s prevent high nucleosome density. We performed gel shift assay with our nucleosomes and 1H6 antibody. In the presence of the excess of histones, 1H6 formed no complexes with DNA, but after the pre-incubation with G4 stabilizing ligand peridostatin, it bound both constructs, suggesting that the control construct contains a hidden G4 in the nucleosome positioning core. To clarify this matter, we next performed band shift assays in a histone-free system. Both DNA constructs interacted with G4 antibodies, supporting the presence of a hidden G4. To locate the hidden G4, we inspected 50-55 base pair fragments of the DNA constructs using fluorimetry assays with a G4-specific light-up probe and circular dichroism spectroscopy. The G4 linker was folded, its mutant was unfolded, and the hidden G4 was found in a fragment A of the mononucleosome positioning system. As a control DNA construct is not fully G4 free, we used additional commercially available DNA control 3, which showed no signs of G4s in a thiafluorine assay. Atomic force microscopy confirmed nucleosome assembly on all DNA constructs. The most common species in the scans of DNA histone mixture were terminal mononucleosomes with linker DNA tails. Free DNA duplexes were also abundant. These knots are presumptively G4s. Atomic force microscopy also revealed some rare species such as D-nucleosomes, disassembled nucleosomes, nucleosome sliding, and G4-G4 interactions in duplexes, kissing complexes. We studied interaction between chromatin remodelers, gene 3 and BRD3, and mononucleosome. Gel shift assays showed that chromatin assembly factors bind nucleosome and the free nucleosomal core DNA, probably including the formation of multi-component aggregates. This was confirmed by atomic force images and particularly evident for G4 nucleosome. We also studied G4 stabilizing ligands BRCA19 and TMPYP4. BRCA19 has poor G4 versus double-strand DNA selectivity, interacted with all DNA constructs, and efficiently disrupted their nucleosome. TMPYP4, a more selective binder, caused only partial nucleosome disruption at high concentration. These results suggest that G4 ligands and other DNA binders may affect nucleosome occupancy. Our results argue that G4 motifs do not prevent nucleosome assembly, while stabilized or intrinsically stable G4s may exclude nucleosomes. Thank you for attention.